Hello, everybody. Welcome back to another day in the life with Mrs. Meyer. I hope you guys are enjoying my little videos. I love seeing your faces when I do the live instruction. I hope to see more of your faces. It's great to see you guys. It makes me feel a little bit like normal. But I'm going to keep going with genetics. Today's assignment is to do a couple of slides. We're going to kind of dive right into what DNA is. So we talked all about heredity, and your heredity is traits that are passed on from generation to generation because of this molecule called DNA, which stands for deoxyribonucleic acid. You guys can see I have the D, I have the N, and I have the A, deoxyribonucleic acid. Will you ever be asked to memorize that? No, but you should know it. And it's organic. Remember back from our biochem, something that's organic is CNH. I don't know if you guys remember when we did organic compounds, but we did proteins, lipids, carbs, and nucleic acids. DNA is a nucleic acid, and that is the fourth thing that I said we were going to study. It's double-stranded. I know some of you guys are like, is this the one that looks like a double helix staircase? And it is. I'll show you some pictures of it. It's organized in the form of genes on the chromosomes. We've done that in previous slides. Remember, those genes are kind of like those lines, and those chromosomes are the X's. And it's because of genetic recombination. The sperm and egg combine to form a zygote. You guys should remember that from sexual reproduction. And then that organism has a complete set of genetic information. You have half the chromosomes from mom, half the chromosomes from dad. So what's the whole job of DNA? Why do we have DNA? What's the whole deal? Well, DNA's job is to tell the ribosomes which amino acids to assemble to make certain proteins. Remember, I keep on telling you, DNA has the code to make proteins. And what are proteins made out of? Amino acids. So different amino acid sequences make different proteins. Makes sense. It's like different ingredients. Different ingredients are going to make a different product. Everything about us depends on the proteins our ribosomes make. Like I said, your hair color, your eye color, your skin color. Everything is a protein. And where do we make proteins? In the ribosomes. Do you see how everything is connected now? It's pretty cool, right? So these are just some pictures or graphics of DNA that you can find on the internet. It is double-stranded. You could see it's kind of got the one strand here, and then it's got the other strand here. This is more of like a 3D model of it. It's pretty cool. And there's some weird letters in here. I'm going to show you all about those. So the genetic code. This structure was discovered by these two guys named James Watson and Francis Crick. They were a professor-student combination. I think James Watson was the professor, Francis Crick was a student. It actually was discovered over here on Long Island at the, why can't I think of it? I don't remember. It'll come to me. But it was found here on Long Island. They wrote a book about it. I had to read the book in college. It was not a very interesting book, but it was a big accomplishment. So they were the ones who discovered the model of DNA, that it looked like a double helix or twisted ladder. These are different words that you might see. So DNA is a polymer. Remember this prefix poly? Poly means many. So basically, it's a long chain of repeating units called nucleotides. So the same structure keeps on repeating and repeating and repeating and repeating. So this nucleotide repeats itself. And the nucleotide has three main parts. It's got a phosphate, it's got a deoxyribose, which is the sugar, and it's got a nitrogenous base, what can be one of four things: adenine, thymine, cytosine, or guanine. It could be A, T, C, or G. So this is a very important diagram. This is one nucleotide. I call it kind of like my house. So I say, this is my house. This is the swimming pool, pool, and this is the driveway. If you guys look at my house, it is a five-sided figure. 5 sides, that means that it's the sugar, deoxyribose. It's a 5-sided sugar. So this is the sugar. This is the pool, because it's the phosphate group, P for pool for phosphate. And this is the driveway, which is the nitrogenous base, which could either be an A, T, C, or a G. So you have your deoxyribose, you have your phosphate group, you've got your nitrogenous base pair, And here it is in a bigger structure. So if we took DNA and we kind of untwisted it, this is what it would look like flat. And do you see here how I've circled? This is one whole nucleotide. It's got a swimming pool, it's got the sugar, and it's got the base pair. So if you look, how many nucleotides are there in this diagram of DNA? Well, you have to look for the same structure that's repeating. So there's one here, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So there are 12 nucleotides in this picture. So over here just kind of goes through what each thing is. Number 1 is a phosphate group. It's the pool. 2 is a bond. 3 is a deoxyribose sugar. It's a 5-sided sugar. Again, you have a bond. You have your nitrogenous base pairs. And then 5 is saying there's a weak hydrogen bond here. It's not held together very strongly, and I'll get into that next week when we talk about how DNA replicates itself. It's not held together strongly because DNA is constantly breaking apart, so it can reproduce itself, basically. So the sides are the phosphate and the sugar. And then the rungs, sometimes we like to compare this to a ladder. So the sides are the phosphate and the sugar, like the side of a ladder. So it's the pool and the house. And then the rungs or the steps are the nitrogenous bases. And they have to go in a specific order. A always has to go with T. G always goes with C. They always go together. So I remember it at Garden City. At Garden City. A always goes with T, G always goes with C. And they are held together by weak hydrogen bonds. So here's just another picture of it. Remember, this is one nucleotide. So if you guys look, here is one nucleotide. It's got the nitrogenous base or the driveway, the sugar, and the pool. So you have to look how many of them are on the screen. It says there are 8 nucleotides on the screen. So you would say this is 2, 3, 4, 5, 6, 7, and 8. So there are 8 nucleotides on the screen. And that's basically what DNA is. It's just repeating itself with this whole different code. And remember, A always has to pair with T, G always has to pair with C. We're not moving. There we go. I'm trying to see where I want to go. Sorry. Okay, so when working with strands of DNA, always group the bases in 3s. So we have all these letters, and they kind of get a lot. It gets so crazy. So what you do is when you're reading a code of DNA, you always just want to read everything in a group of 3. So A, T, T, G, T, T, right? These don't really look like groups of 3. I don't know why. But these are codons where everything is 3 bases long. I don't really love this picture very much. Sorry about that. How to find the complementary base sequence in DNA. So A always goes with T, T always goes with A, G always goes with C, C always goes with G. So if you had to find the complementary base sequence, remember we put them in groups of 3. ATT, TTG, CTC, ATC, GTG, CCC, ATC. So what you would do is you would match up the A. What goes with A? T. What goes with T? A. What goes with T? A. So our first codon would say TAA, and then so on. We would do so much practice with this if we were in school, but right now we're not. So what would the sequence of the complementary base strand be? So if I gave you TGAC, what would you put in the complementary side? They call this the opposite side the complementary side. So you have to think in your head, what goes with T? What goes with G? What goes with A? And what goes with C? See if you can figure it out before I put up the answer. So a complementary would be ACTG. This is just a little video recap of what I just did. And this is all I have for you guys today. We're going to kind of go through a little bit more DNA stuff on Monday. Maybe I'll try some questions with you guys. And then I will see you guys again on Tuesday. I hope you guys have a great weekend. If you need me, I'm here. You can message me for any reason or email me. Okay? Take care.